Good afternoon, everyone, and welcome to the front row. My name is Jamie Williamson. I'm a professor here at the Scripps Research Institute, and we're really pleased that all of you could join us here to hear Jeff Kelly's talk about neurodegenerative diseases and how to treat them. I would also note we have a virtual audience, and we have, I think, well over 1,000 people that have tuned in to watch the proceedings. And for all of you in the ether out there, we would encourage you to come to campus next time. It's a great way to meet people and meet the faculty who are speaking and find out a bit more about what's going on here at Scripps. And I would be remiss if I didn't point out the large digits on the side of the stage. We're approaching our 100th anniversary, and So there's more to come on this, lots of activity to celebrate the science and prepare for the next 100 years. So today's subject is neurodegenerative diseases, and I mean, I've got to be honest, this is, as we all age, I'm aging too, and it's becoming more and more on my mind what's going on. And there are a variety of these kinds of diseases that you're all probably well familiar with. There we go. And so the problem with these diseases is there's not really any cure, and the current frontier is to try to discover therapies that will mitigate the progress of the disease, and we're going to hear a great story about that today. But one thing I do want to point out is that philanthropy plays a big component in funding research such as the medicines that you'll hear about today. The primary mission of the front row is really to get our story out to the world and the community, but we don't want you to think that we're not interested in how you might participate with the Institute. And the one thing I would say is even a small gift can make a huge impact to someone that's a graduate student, and these projects that you're going to hear about started out with graduate students. Now, there are currently 12 drugs that are approved for treating neurodegenerative diseases, and we're just proud that 4 of those have actually come out of Scripps. So there's Ernie Beutler developed a drug for MS, and you heard, if you tuned in last month, you heard Hugh Rosen talk about ozanimod, and Stuart Lipton has a drug just approved for Alzheimer's, and then today we're going to hear about Vyndaqel and Jeff Kelly's story. And just to illustrate the impact that this medicine has had, we have a short video.
I believe I wouldn't be here, and I think we agree. I was initially diagnosed in the fall of 2018. The name of the disease is amyloidosis transthyretin with cardiomyopathy.
Tafamidis very much extends lifespan and quality of life in people who have these life-threatening disorders.
So from my standpoint, it's changed my life, and it's really given me a new lease on life. I still get to spend time with my grandkids, which couldn't do without this. So that's a big one.
So this is one story, and safe to say that there have been tens of thousands of patients who have benefited from this medicine. So I want to say a little bit about Jeff Kelly. So Jeff and I have known each other for quite a while. I dug deep into the archives. I found this picture up in the upper left, and this picture is from 1991, and Jeff Kelly and I were both awarded the Searle Scholar. This is a junior faculty award that is a great career boost. But what I really enjoyed about this picture, this was 6 of us, but the person on the far right is Reza Gadiri, who's on the faculty at Scripps, and he has been ever since 1991. That's me, the second from right. And at the time I was at MIT, and then Jeff is third from the left. So neither of us had any clue that in 1997, 1998, when we came here, that we would all be together at Scripps. But it's just a fun memory to think about. And I would also observe that, that all of us have less hair now than we did. But I just started from a higher baseline. Okay, so without further ado, I'm going to introduce Jeff, and he's going to tell you a little bit more about how he came to be here at Scripps and how he came to discover tafamidis. At the end, I'm going to jump back on stage, and we'll have a Q&A. I'll do a little discussion. You can raise your hand, and we have microphone runners that have a Jedi lightsaber that will be visible to me, so we can we can take questions from the audience. And if you're virtual, you can type a question in at any time during the talk, and they will get relayed to me, and I can take questions from the virtual audience as well. And I just want to remind everyone that neither Jeff nor I has an MD. We are not physicians, and we can't give personal medical advice, so we're happy to entertain any questions, but just keep that in mind, that we can't provide medical advice. So really, without any further ado, Jeff, please join us.
Thanks, Rob.
So good afternoon, everyone. It's a genuine privilege to be here. My lab had the good fortune to discover the first drug that slows down the progression of a neurodegenerative disease. And it was a long period of time, about 8 years, between the approval of that drug and the next one. And the community during that period got very negative and said, well, transthyretin amyloidosis is an anomaly. But as you'll see today, it's actually pretty representative of all the neurodegenerative diseases. And what I'm going to try to do today is to take you through how we had the good fortune to discover the first drug and where the others have come from. And San Diego is featured very prominently in the roughly a dozen drugs that we now have for slowing neurodegenerative disease progression. You saw the people from Scripps that have contributed, but Ionis Pharmaceuticals up the road have contributed substantially, and also I'll mention some folks at UCSD. And lastly, before I dive into it, let me just say that this is a team sport. The faculty are the coaches, and the people who really make everything happen are the graduate students and postdoctoral fellows. And Evan Powers, who's sitting over there far left, was a postdoctoral fellow, and he single-handedly discovered tafamidis. He wrote the structure out, wrote an NIH grant on it, and it actually worked as he anticipated. So, a little bit about me, very little. So, I grew up in a small town in western New York called Medina. There are probably more farm animals than human beings there. I was one of 4 children, grew up in a really supportive household. And probably the most meaningful thing I did when I was a kid is I worked on various farms. And quickly I learned how to fix things, because if you didn't, the owner of the farm was very upset with you if the field wasn't plowed. No matter what went wrong, you had to fix it. So that was a really good work experience for me. And I would say that I never considered myself a scientist until I was in college or interested in science. I was probably curious, much like my daughter Olivia, who views her necklace with a locket as a great place to put roly-polies and do experiments, right? And I think all of us have that curiosity when we're kids, and the most important role of a parent is not to dial that out of the kids, of course. And Mr. Tierney was my high school chemistry teacher, and I didn't think that I had a lot of aptitude in chemistry, but he kept reminding me that I did. And it wasn't until I got into college at a small SUNY school south of Buffalo that I realized I really liked chemistry. And my sophomore organic professor came up to me and said, you should bail on engineering and become a chemist. You actually have aptitude at this, and there aren't many people who do. And so I didn't do that right away, but eventually I did change my major and focused on undergraduate research, which was a wonderful experience. So I moved to Scripps in 1997. Richard Lerner, I had the great fortune of meeting him, and he recruited me here. And this institution has really shaped the way that I think about science in a very positive way. I think, how does somebody like me get interested in neurodegenerative diseases? It's not obvious, right? I was trained pretty rigorously in chemistry, and when I was a postdoctoral fellow at Rockefeller University, I was quite interested in how proteins, which I'll define in a few minutes, adopt their proper shapes. And while I was reading about a protein I could study, I came across these so-called amyloid diseases, which I knew absolutely nothing about. But I realized after a few days in the library that this is what I was going to do with my life, because I thought I understood these diseases, and I didn't think the people who were writing about them did. And fortunately, it turned out to be that way. Okay, so Jamie already outlined some of the major dementias. So Alzheimer's by far has the largest number of patients. I should probably use the online pointer. Let's see, excuse me just a second. There we go. There we go. So there are at least 50 million patients worldwide that have Alzheimer's disease. And just a couple years ago, I couldn't have mentioned that there was a disease-modifying therapy for this disorder, but now there is. And I personally think it's really good, and I'll show you the data towards the end of the talk. These diseases are all dementias, so you have an impaired ability to think and remember that's serious enough to impair the quality of daily life. Parkinson's disease is the disorder that affects the second most people. And transthyretin amyloidosis, the disease we developed a drug for, is the third largest amyloid disease. And to be perfectly frank, when I started working on this 35 years ago, nobody knew that that was true. So turns out that the genotype that almost all of us have, which is normal on both of our gene copies, also leads to autonomic nervous system dysfunction and cardiomyopathy as we age. And I'll tell you a little bit more about that disease in a few minutes. So in all these diseases, we lose neuronal function first, and then we lose the actual neurons themselves. And as you may know, neurons are quite unique cells in that they're highly extended cells. They're quite polarized. They have a cell body, or soma, and then these tree-like projections are called dendrites, and they receive signals from the nearby neurons. And if the signals are strong enough, an action potential propagates down the so-called axon. Axons can be anywhere from very short to meters in length, depending on which neuron you're talking about. And then chemical signals are released at the so-called synapse to interact with receptors on the next neuron. And so you have this incredible connectivity. The unfortunate thing, I suppose, is that neurons don't easily regenerate. And so we're born with about 75 billion neurons comprising our brain, and we're going to die with fewer. So the connectivity in the brain and the redundancy is immense. So we lose neurons all of the time. Generally, that doesn't cause significant cognitive dysfunction. But once you start to lose 25 or 30% of the mass of your brain, that loss of connectivity can become really problematic. And those are the phenotypic things that we see in the clinic, dementia and so on. Now, one of the things that we've understood about these diseases over the past few years is that they not only destroy the central nervous system, but they also destroy the function of many organ systems. For example, Parkinson's disease often manifests as a GI deficiency about a decade before it becomes a central nervous system disorder. Very much true also with the transthyretin amyloidosis. In Alzheimer's disease, organ dysfunction appears in parallel with dementia. So if you don't remember anything else about this talk today, remember that these are diseases of protein shape or conformation. So proteins typically are very small. They're 2 to 3 nanometers in diameter. And remember, a nanometer is about a billionth of a meter. So these are really small things that you need an electron microscope or the like to see. When proteins change their shape and undergo aggregation, they can become visible to the naked eye sometimes, but easily visualized in a light microscope, become much, much larger. These things I'll define later. They're called amyloid fibrils, and it's the reason that all these diseases are called amyloid diseases. And of course, when I started working in this field, the notion was that these amyloid fibrils were the main drivers of pathology. To a chemist, that made no sense. These were deeply buried in the extracellular space. They really weren't amenable to very much biology or pathobiology. So as you'll see, we took a risk and decided that it wasn't logical that these were the main drivers of pathology. So what is a protein? So, of course, the genome encodes proteins through the intermediacy of messenger RNA. So transcription gives RNA, and a process called translation mediated by the ribosome that Jamie Williamson studies leads to proteins. The way that I think about proteins is that you can think about them as an unlatched pearl necklace, where the pearls can be one of 20 different types, and the individual pearls have modest affinity for one another. And that modest affinity allows the pearl necklace to spontaneously collapse on itself to form a unique three-dimensional structure that's matched to the sequence encoding that protein. That process is called protein folding, and that juxtaposition of functional groups is absolutely critical for the function of proteins. You can get very interesting catalytic activities and so on that allow our physiology to take place. But protein folding is never complete, and it often competes with another process called protein aggregation, and that's shown on this slide. So conceptually, if you think about a protein as it comes off the biosynthetic machinery, namely the ribosome, as an extended pearl necklace, then because of the affinity of specific amino acids or pearls for one another, the protein adopts a specific shape. It can also adopt the wrong shape, and there's quite a bit of machinery in our cells, at least 2,500 genes that have evolved to deal with these problems. The most challenging problem is that these amino acids can also satisfy their proclivity to self-assemble or to interact with one another through self-assembly. And this process is called protein aggregation. And the thinking is that having the wrong protein conformation or a misassembled form of that protein is the main driver underpinning neurodegenerative diseases. To me, this was clear from the genetic literature 30 years ago. People made a lot of observations that suggested that it was this competition between normal folding, misfolding, and aggregation that was leading to these disorders. So why is aging the main risk factor in these maladies? It's probably because up until a certain point in aging, we can do a pretty good job of getting rid of the misfolded population and the misassembled population through processes known globally as cellular degradation. And those processes are mediated by the proteasome and the lysosome, macromolecular machines that very efficiently degrade proteins back into their individual amino acids or pearls. And the thinking is that when we age, and in fact there's quite a bit of evidence for this, if you're unlucky, then your protein degradation machinery doesn't work as well as it did when you were 20. And if you have additional risk factors, that can lead to various neurodegenerative diseases. And there's really good genetic evidence for that hypothesis, as well as pharmacologic evidence that I'll take you through today. Now, I mentioned earlier the idea that these diseases are called amyloid diseases because the particular shape that proteins can adopt in these really extended manners allow individual proteins shown by these different colored arrows to self-assemble with one another, generating structures that can be comprised of 1 million copies of one protein. And this particular shape is referred to as an amyloid fibril. These diseases today are generally diagnosed with small molecules that localize to the fibrils. These are PET imaging agents, and that's how physicians often tell you if you're at high risk for Parkinson's or Alzheimer's disease. And if you have genetic predisposition and symptoms, the PET agents are enough to diagnose the disorder. Now, the reason that we weren't too excited about amyloid fibrils being the main driver of these disorders is because they were buried in the extracellular space and coated with a variety of other macromolecules, and hence they were basically unavailable. It's a little bit like burying nuclear waste. Clearly it's not good for you, but if it's 5,000 feet below the ground, you can have a house close by with low risk. What we thought was much more risky was the notion that proteins in their normal attempt to fold spontaneously, and often with biological assistance as well, can form other type of structures that are incorrectly folded and or assembled. And we hypothesized that maybe these were the structures that were actually driving the process of neurodegeneration by engaging both in normal biology that they shouldn't and in destroying things like membrane architectures that are so critical for allowing cells to perform normal functions. So the concept that we came up with was rather simple. The notion was that if you have a protein that adopts a well-defined three-dimensional structure, can you fashion a small molecule that would stabilize that structure and prevent it from undergoing conformational excursions that can happen after the protein actually folds, since these processes are reversible very rapidly often. So the notion was that you would stabilize the protein in its proper shape. That would still allow the protein to perform its normal biological functions, but then you would basically prevent everything else from forming, with the exception of amyloid fibrils, which shouldn't be blacked out here, but a very able assistant made this slide for me today, fortunately, or I have very little to talk about, but that's the only mistake, right? Because amyloid fibrils are deeply buried. They're not going to go away if you stop the process of biosynthesis from producing new aggregates. But all of these other things are mobile enough that the normal degradation machinery are going to get rid of them. Now, to be fair, the pharmaceutical industry thought this was a really crazy idea because the thinking was so strong that amyloid fibrils were the main drivers. So what we know now is that this ensemble of protein aggregates engages both normal biology, compromises cellular architecture, and one of the main problems with neurodegenerative diseases is that the transcriptional programs, that is, the proteins that are expressed that determine a neuron being a neuron and an endothelial cell being an endothelial cell, get really screwed up in these neurodegenerative diseases. So the cells lose differentiation. That's clearly a huge problem in our physiology. There's a lot of other things that happen too that I don't have time to take you through today, but suffice it to say, these diseases are probably best described as death by 100 cuts, death of neurons initially, right? And it's because neurons are quite sensitive and they don't regenerate, and so ultimately they drop out of the brain. Now, the transthyretin amyloid diseases, we became very interested in them because they're one of the few amyloid diseases that are so-called cell non-autonomous. So what does that mean? It means that the tissues that are destroyed, like organ systems, the autonomic and peripheral nervous system, as well as the central nervous system, they don't actually make transthyretin. So transthyretin is a protein that's secreted into the bloodstream or into the cerebrospinal fluid that bathes the brain. And the notion is that when this protein aggregates while it's in circulation, it can destroy tissues that don't make the protein. And we thought that was a big advantage over what people were struggling with in Parkinson's and Alzheimer's disease, wherein A beta and tau, the proteins that destroy the brain regions in Alzheimer's and Parkinson's, also make those proteins, which makes it very difficult to figure out how the pathology works. Okay, so this protein is made and sent into the blood by the liver, and it's made and sent into the brain by the choroid plexus. So because the concentration is much higher in the periphery, this is kind of a rare amyloid disease where the peripheral manifestations occur early as 20 or 30 years of age, depending on if somebody inherits a mutation or not. And then as the patients get older, they develop central nervous system pathology, namely dementia and cerebrovascular bleeding, very analogous symptoms to what happens in Alzheimer's disease. So the transthyretin protein has a very defined shape. You can see these little arrows just represent the pearls in an extended shape or conformation. And the fold is quite intricate, and that's generally true as protein folds go. This protein is comprised of 4 identical subunits that are differentially colored here. And in your bloodstream, there's 3 forms of this protein. One that has a little bit of thyroid hormone bound, that's only a couple percent. 50% of it doesn't have any other molecules bound to it. And then about half of it carries vitamin A via this red protein, which is called retinol binding protein. So it's this form of the protein that doesn't have any ligands that can dissociate, misfold, and aggregate. And we knew that that was the process by which this protein undergoes a shape change because probably 30 graduate students worked on this mechanism over the course of the last 30 years. So we learned that the cell biology really doesn't have a big role. Cells can sometimes be very picky about which proteins they secrete. But it turns out that transthyretin gets secreted even when it's mutated and unstable. So the cellular quality control machinery doesn't have a very big role to play in protecting us from these diseases, especially in the brain. What we found out that was really critical and was enabling for drug discovery was the notion that the protein ultimately undergoes a shape change and it aggregates very efficiently. But that's not rate limiting. What's really slow in the process is dissociation of the native structure that allows this shape change. And that was a huge advantage, of course, because I already told you that this protein, about 1% of it binds thyroid hormone. So there are 2 binding sites that are already there. and they're largely unoccupied. And so since this is the slow step that allows aggregation to occur, we rationalized that we could make it even slower by occupying one or both of the small molecule binding sites that weren't occupied. And when Ted Foss was a graduate student, he did some elegant experiments showing that this protein comes apart about this interface that binds the two small molecules. So the notion was to make a small molecule that could be taken orally, ideally once a day, that stabilized the protein towards all of these other structures that could form. And we argued quite aggressively over the years that this was a conservative approach, because we didn't have to know which of all the structures that this protein could form that were not native, which were driving pathology. Now, the really smart people, of course, countered that argument and said, well, if it's amyloid fibrils, you guys are hosed, because you're not going to change the world. You're not going to change their concentration at all, and therefore, this is unlikely to work. Well, that turned out to be not true, fortunately. Now, we call these drugs kinetic stabilizers because they increase the barrier associated with tetramer dissociation. So all processes in chemistry involve barriers. Sometimes the barriers are very small. That means that things happen very fast. And if the barriers are very high, things happen very slowly. The nice thing about small molecule binding, so long it's selective to the normally folded form of the protein, you can stabilize and lower the energy of the native fold. That increases the activation energy, barrier. So much so that when the drug is bound, the protein cannot dissociate and it cannot form aggregates. It can only come apart in the lysosome where it's supposed to be turned over and degraded back to the amino acids. So I mentioned nobody had ever done a clinical trial on any amyloid disease when we tested this candidate compound, tafamidis. So the clinical trial that we decided to pursue was the brainchild of Teresa Callejo, a physician in Portugal who takes care of individuals who have mutations in transthyretin that lead to early onset polyneuropathy. And we convinced the Food and Drug Administration that this was the best trial to do because we really understood that disease very well. And they agreed that that was really a brilliant thing to do. And we completed the trial using this neurologic impairment score. And the way that clinical trials work, in my opinion, are crazy. John might or may not agree with me, but you have to predict the answer pretty accurately. And if you're off such that your statistics are p-value 0.06 instead of 0.05, the drug fails. So this trial, as you can see, there was a very clear slowing of disease progression with tafamidis versus placebo. But the FDA didn't approve this drug because the statistics were 0.056, I think, which is really insane. But okay, we can argue about that later. Fortunately, the rest of the world approved the drug, and very quickly it became clear that the drug worked very well in the real world. Now, the most important thing we learned from this trial was that in a demographically very similar group, that is the placebo group versus the drug candidate treated group, if you delayed treatment for 18 months, then only roughly 46% of the population benefited from the drug. If you started treating them only 18 months earlier, then it was something like 60%, or even 70% in some situations benefit. So huge benefit from early diagnosis and early treatment. This has been borne out in every single neurodegenerative disease trial since. Early treatment is absolutely key to having the maximal clinical benefit. Now, tafamidis eventually got approved in the US when we decided to do a cardiomyopathy trial. And at this point, Pfizer bought the drug from the company that we started to do the first trial. So when we did the first trial, it wasn't appreciated that transthyretin cardiomyopathy was a major disease. The reason that tafamidis sold $1.1 billion last quarter has nothing to do with neuropathy. It has everything to do with there are a lot of cardiomyopathy patients. So the cardiomyopathy patients have the same genotype that all of you have. And why roughly 10% of men develop cardiomyopathy when they get older, we don't know, other than it involves protein aggregation. But we don't know why their wild-type protein aggregates and those that are disease-free do not. Now, this trial basically looked at frequency of cardiovascular hospitalization and all-cause mortality. And basically, you don't see a huge difference between the treatment group and the placebo group. But the statistics here were, I think, 0.00016, so highly significant because we enrolled a lot of patients, and big pharmaceutical companies are very good at this. But you can see, again, this huge advantage of being on drug versus placebo. And this is, every time there's a downward blip here, this is an unfortunate patient death. So you want these curves to be flat. But they can't be flat in a clinical trial because you have no data. So you have to pick patients that are pretty sick. But you can see quite clearly that being on continuous tafamidis is a huge benefit, especially as you go further down the aging process relative to being on placebo and then switching on to drug. Again, early treatment is absolutely critical. Now, the early Alzheimer's trials with successful agents look something like this. And even though we admire immensely physicians, most of them have never seen a clinical trial and never designed one. And most of the Alzheimer's community concluded that the data you'll see in a few minutes really wasn't very good. When you get separations like this and then you go into the real world and treat patients much earlier, now the people on tafamidis with cardiomyopathy basically look like this, right? They're barely declining, and that's because you can use the drug early. So you have to be very careful about over-interpreting clinical trial data. Now, I mentioned earlier that transthyretin amyloidosis also causes dementia. And fortunately, and certainly not by design, tafamidis gets into the brain quite well. And so people that were treated 15 years ago are still free of dementia. So we're pretty confident that this drug has significant brain exposure to prevent neurodegeneration. But that was purely luck because we didn't know that dementia was a serious factor in these diseases until we were able to treat the periphery and then realize for agents that didn't get into the brain that there was a CNS problem. That, I shouldn't have said agent. Historically, people with mutations were actually treated by liver transplantation, ironically, because the liver, remember, secretes the protein. So if the liver secretes a mutant protein that's unstable, you can do a liver transplant to fix the disease. The problem is that the risk of death in these patients from liver transplantation is about 10%. So you take somebody who's apparently healthy in a small town that has these mutations, and nobody in the town will get a liver transplant after that because the assertion is, hey, this person was 30 years old, they were perfectly healthy, and they died of this procedure, not of the disease. And that was true, but had they not taken that risk, they would have been very sick a decade later. Okay, so that's basically how we figured out that this disease eventually manifests in a dementia. Okay, now let's skip ahead. So tafamidis was approved in the rest of the world by the regulatory agencies in 2011. It was approved in the US in 2018 for cardiomyopathy. So, no, that's not right, is it, Evan? What year was it approved for cardiomyopathy? 2019, that's right. So, Ionis up the road has developed drugs that deplete the transthyretin protein by degrading its messenger RNA. Those drugs also prevent the aggregation of newly biosynthesized transthyretin. They do not clear amyloid fibrils. So we call all of these drugs that don't clear amyloid fibrils category 1. So you have stabilizers, and I didn't mention this today, but for patients who can't afford tafamidis or are in a country that doesn't support its use, we discovered that the Merck nonsteroidal anti-inflammatory drug diflunisal is actually quite a good stabilizer, and it's pennies a day. Now, the reason it's not widely used is that compromised renal blood flow, which is a problem in this disease. So these patients are a little harder to manage, and the physicians, if at all possible, like to use tafamidis. Then I mentioned Ionis being a big player in making drugs. So this was the first drug that slowed down ALS made by Ionis. So this is a particular form of ALS, though, a hereditary form that results from the misfolding of a protein in the cytosol that is the bowels of the cell called superoxide dismutase. So there was very strong evidence generated from Don Cleveland, who's at UCSD, that misfolding of this protein was driving this form of ALS, and Ionis made a drug that destroys the messenger RNA, and this trial actually failed. You can see that there's a clear difference between the treatment group and the placebo group, that is, the placebo group declining more rapidly, but the difference wasn't as predicted, and so the trial failed. But they were smart enough, as is usually the case, to have an extension period where everybody goes on drug. And you can see clearly that those individuals treated with drug benefit immensely relative to those treated with placebo and then rolled over to drug. So this was approved for this form of ALS in 2023. Now, the other big advance that has taken place are the development of monoclonal antibodies that are injected into our bloodstream. And after decades of effort, people have figured out how to get them to cross the blood-brain barrier. These are antibodies that clear amyloid fibrils, okay? So what I supposed about amyloid can't be precisely true, because if you clear amyloid fibrils, and these drugs do it very well, the progression of Alzheimer's disease slows down. And you can see that in this study. This isn't a huge separation, but I think this trial is roughly 1,500 people. So the statistics are very clear that being on drug versus placebo slows down the progression of Alzheimer's disease. And of course, as I mentioned earlier, you have to pick individuals that are sick enough so they decline, so you can see a difference if the drug really works. Now there's evidence actually that in early stage Alzheimer's disease that this antibody, lecanemab, made by Lars Landfeld, an academic scientist in Sweden, probably, if not stops, it dramatically slows the progression of early stage Alzheimer's disease. And where it's particularly effective is when the patients don't have a very high tau load. And if you're curious about that, we can talk about what that means afterward. Now, recently, just a few weeks ago, an antibody made at UCSD by Eliezer Maslia, and after many years sold to Roche, the clinical trial they ran for Parkinson's disease failed. But upon reanalysis of the data, there's quite compelling evidence that for rapidly progressing Parkinson's disease, this drug probably works. And so there's going to be a lot of motivation now to more critically evaluate this antibody. So another San Diego UCSD contribution. Okay, so why does clearing amyloid fibrils slow down the progression of these diseases if they're buried in the exterior? I still think that everything I told you about amyloid fibrils is true up to a point. So about 5 years ago, this woman, Alison Goad, who's a scientist in New York City, kind of reevaluated a lot of human genetic data on Alzheimer's patients, and she came to the conclusion that a lot of risk factors that hadn't been well understood in Alzheimer's disease affected this class of cells called microglial cells in the brain. So these are cells that can attack non-native protein conformations and destroy them in organelles called lysosomes. It's still not at all clear how these cells take care of something that's 100 times bigger than they are, but people are trying to figure that out. It's absolutely clear, though, that these drugs that are monoclonal antibodies recruit these cells to the amyloid fibrils, and you can see in clinical trials by PET imaging that within a year the fibrils are gone. So recruiting these cells work very, very well. So what we think is going on, I'll leave you with a hypothesis that needs a lot of work, but we think what goes on in these diseases is that due to an inflammatory trigger, the microglial cells start to attack the amyloid that has been buried and dormant for decades in some cases. The problem with this process is that it's incomplete. So you now expose the brain to a lot of misfolded protein, and the problem is that this can template or nucleate aggregation in nearby cells. And we've known that mechanism for years because that's how prion diseases are infective. But the thinking was that that was unique to prion diseases. But I think now there's abundant evidence that while we don't get Alzheimer's disease from eating something, we can get it from one or a few rogue cells that start to spread these fibrils. And there's quite a bit of evidence in the literature for that. So the notion is that you start making a lot of amyloid that's in the extracellular space. That allows cell-to-cell passage. And then ultimately what we see in these diseases is not exactly what people thought 5 years ago. So the notion was that in Alzheimer's disease, it's aggregated A beta and tau, and that's it. Now we know these patients are loaded with alpha-synuclein aggregates that heretofore have been associated with Parkinson's disease. TDP43 is another aggregation-prone protein that's aggregated in many neurodegenerative diseases. So we hypothesize that when the immune system starts to attack amyloid, now these naked fibrils in the extracellular space can start to template the aggregation of proteins that wouldn't have ordinarily aggregated without these templates being around. So, that to me seems to explain how it is that you can have drugs that work through downregulating the proteins capable of aggregating, as well as drugs that work through knocking down amyloid fibrils. And we'll see. There's some really beautiful Parkinson's data that's still preclinical that shows that if you alter the surface of these amyloid fibrils, you can also prevent the progression of the disease. So I think there's something to this notion that the surface of these proteins can be driving the pathology. Anyway, so the genetic data is abundantly clear that protein aggregation causes neurodegeneration. I mentioned the situation that people became very skeptical because of all the failed clinical trials in Alzheimer's disease. But now we have a dozen drugs that work for these disorders. They all more or less work through preventing protein aggregation. So I'm confident that, in the next decade, we're going to have combinations of drugs that are incredibly effective in further slowing, if not stopping, these maladies. So it's been a long 35 years, but I'm really proud of where the field is now. I think we're finally delivering on all this basic research that we did and translating that into medicines that really work in the real world. So reason to be optimistic. And if you don't, the other major take-home is that if you think you might be developing one of these diseases, don't procrastinate. Early treatment is absolutely key in slowing down their progression, and now there are drugs that do that. Thank you very much.
So we'll have some questions here, and I've got a few from the virtual audience. And if you have a question here in the real audience, just stick your hand up, and one of the Jedis will find you. But let me maybe start with a question. So, and this is just kind of historically amusing to me, because I used to be on your thesis committees of your students. And I remember, seeing these transthyretin binding molecules. And you said Evan wrote down, so how different is the molecule that you guys first made from the one that's clinically approved? Because often there's like a long path of medicinal chemistry and optimization, and so how close did you land?
Ultimately, we made about 1,000 small molecules in this program. And it was mostly graduate students and postdocs that made those molecules. And we designed those molecules on the basis of an X-ray crystallography-based structure, which we now know from cryo-EM isn't quite perfect. Mm-hmm. It's really close. And so, had we had the perfect structure all along, we might have had to make 500. X-ray crystallography doesn't help you completely because thermodynamics are very complicated and you can't predict binding constants, and you need strong binding to have an effective drug.
So here's a virtual question. So there's other diseases that you didn't touch on, like there's frontotemporal dementia, there's Lewy body disease, and there's all kinds of dementias. So are they all due to protein misfolding, and are they the same proteins or different proteins?
Well, at least all the major ones are very likely due to protein misfolding and aggregation. So Lewy body dementia is a form of Parkinson's disease. The other disorder you mentioned is a tauopathy. So tauopathy is also a component of kind of the second stage of Alzheimer's disease. And so there's a lot of people working right now on developing drugs that prevent the misfolding of tau. So I'm really optimistic that, in the next 10 years, we'll have more than one drug for all the major neurodegenerative diseases.
Right. I'm missing any? Okay, we have a question over here.
Thank you. And I also appreciate you giving all this credit to your graduate students, because I know some professors wouldn't do that, so that's great of you. But I wanted to ask, along with this great work you've done with new treatments, are there also much earlier diagnostic treatments in terms of the DNA, or what should I be getting now? Like, I'm 67 or something. What should I get now? Can I get a DNA treatment test or something like that?
Yeah, that's an excellent question. So there's now very well-funded efforts that have been underway for some years now coming to fruition for various early diagnostic methods. So they're getting better literally by the month. And so I don't think that's ultimately going to be a problem, but it is today. Today we still use PET imaging by and large for amyloid. The problem with PET imaging, it's expensive. Now there are many people developing blood tests, and those are starting to be approved by the regulatory agencies. Most of them are based on protein aggregates circulating in the bloodstream and very sensitive ways of detecting those aggregates.
That was a good question. You stole one of mine. So Jeff, you explained very clearly how tafamidis inhibits the formation of fibrils, but the fibrils don't dissociate. But then at the end, you were talking about how the microglia attach those, or detach those and dissolve the fibrils. So has anybody done any clinical trials, or is there any interest in it, in a synergistic combination between the small molecule therapy to prevent aggregation and the antibody therapy to dissolve fibrils?
Yeah, so there are numerous clinical trials in a wide spectrum of neurodegenerative diseases underway that are looking at more than one agent, including antibodies in combination with small molecules. So Ionis right now is looking at the combination of their messenger RNA lowering drugs with our stabilizer. So that trial will read out, I guess, in about a year.
Great. So one of our, sorry, we have a question here. Yes.
Hi.
There's one right down here, too.
Yeah, okay, good.
Sam Armstrong.
Oh, okay, gotcha.
A question, when you were talking about the optimization of the small molecule and how it took all this time, what's your impression of AI and how it'll change all that?
Yeah, well, the problem is I can barely spell it. I'm learning very fast. I do think it's going to have an immense impact. I mean, there was just a paper published in Nature last week from the same commercial organization that figured out how to predict the three-dimensional structures of proteins computationally, saying that they can now design ligands for proteins. A lot of people are still very skeptical of that claim because they didn't put the code in so that people could actually test that hypothesis. But I do think that computational methods are immensely useful. So we just made another kinetic stabilizer for a disease called light chain amyloidosis that unfortunately is both a cancer and a amyloid disease. And as opposed to 35 years, the computational methods allowed us to do it in 5. Mike Petrossi is here in the audience, and he did some amazing medicinal chemistry that was still pretty labor-intensive, but the computational methods helped us a lot.
Yes,
We'll
sir.
be in the
Down in
clinic
front,
pretty soon with that.
question here.
First of all, thank you for your presentation. You've given me amazing information I didn't have before. I am wild-type cardiac amyloidosis. 2017 was my diagnosis. I was in the Stanford study from then on till it went public in 2019, and the only thing that changed then was that they were charging me a fortune for the drug. I have several questions. The first is that you said that the microglia could be used to actually reverse the aggregation of amyloid fibrils in the affected organ. Have you any form of testing at this point to see how much you've pushed the disease back? Because in my own case, my own physicians have been unable to tell me if there's any, if I'm getting better or getting worse or at what rate, anything like that.
Yeah, I mean, it's still early days for understanding the long-term benefits of these drugs and the change in the rate of progression. So, physicians are generally appropriately very cautious. You're at a great center, by the way. There are several clinical trials underway right now in transthyretin amyloidosis, light chain, and the other major neurodegenerative diseases looking at amyloid clearance, say in combination with a stabilizer like tafamidis or a messenger RNA lowering agent. So I think, certainly in the next 3 or 4 years, those trials results will be known.
Second question, this is more, there's a big worldwide organization for cardio amyloidosis. And the function is primarily to bring in research and to educate the medical community on how to diagnose and treat the disease. I have never, I tune into all the lectures, but I've never seen any connection with diseases of the brain. And it would seem to me, based on your research, that this organization should be connected to the myriad of organizations involving research into diseases of the brain.
Yeah, your point's extremely accurate and well taken. I think historically, the Alzheimer's people were siloed and didn't talk to the Parkinson's investigators, who didn't talk to the transthyretin amyloidosis people. There were a few people who saw the common threads and were pushing them, but those people were largely not listened to. I think the situation has dramatically changed now. And the common features of these disorders are absolutely clear. And, people argued with me 5 years ago that, transthyretin amyloidosis patients don't have dementia. And I said, okay, give them the Alzheimer's mini mental exam, and sure enough, they failed it, right? Those patients who weren't treated. And so I think people start to appreciate it, but it takes a lot of evidence to change key opinion leaders' opinions, who are often wrong, unfortunately.
Yeah. Thank you so much.
Well, thanks for giving us a plug for why you come to the front row. You get key information.
One last question.
Oh.
Tafamidis is incredibly expensive.
It is.
About $176,000 a year right now. Even with good insurance, I pay a fortune at the end of the year for the drug. And you mentioned a drug that was pennies a day, but it had some problems with attacking the urinary.
No, it restricts renal blood flow. So cardiologists in particular do not like to use that drug for that reason, because people who have there's physicians here who should speak up, but I think Rob Russo's here. He could probably comment on this, yeah.
Thank you.
Yep. Okay, I think, Jeff, we're coming up on the hour, and so I just want to maybe finish up and ask you one last question, which, this...
Can I mention something, though?
Sure.
I mean, the reason that my lab developed small molecules is in 2029, tafamidis will be 50 cents a day. That's the advantage of small molecules. Biologic drugs, they'll go down when they go off patent, but they never become inexpensive. So Pfizer probably spent $1 billion on the cardiomyopathy trial. So, I'm not going to argue why they priced the drug where they did, but they did it because they could.
So the arc of this story was decades, and so what are you working on now? So what's the next drug to come out of your chemistry brain?
Yeah. So so what I should have mentioned, and I failed to mention in my talk, is that every single drug we have right now targets a particular protein. And as we learn more about these diseases, we realize that while there is a dominant player in driving neurodegeneration, there are other proteins aggregating in Parkinson's disease and Alzheimer's. And so it would be nice to have a strategy where we speed up and make the degradation processes that I mentioned are the risk factor for these diseases. So those are the drugs we're working on now. to tap the biology to make it more efficient so that whatever aggregates you're forming are not going to persist.
Great. Well, we look forward to having you back. Now, so let's let the next front row. So we're taking a month off, so the next front row is going to be Mike Ballong, who's one of our young faculty at the forefront of drug discovery, and it's July 17th is the next front row. So if you're virtual, we invite you to come for real. For those of you here, there's going to be a reception out in the courtyard there. And I do want to draw your attention, we have a gold table, which will be staffed by people from the advancement department. So if you're interested in becoming more engaged with Scripps, stop by the gold table. But otherwise, thanks a lot, Jeff, and I will see you in July. I'll be in the front row.
Awesome.